Welcome to Biology My Passion. I am Soumya Harikrishna. Now we are discussing the chapter Biomolecule. We discuss the analysis and also we discuss the amino acids, which is a biomicromolecule. Now we are going to discuss another biomicromolecule, that is lipids. Lipids or fats are very common term to us, and they are basically organic compounds which are greasy or oily in consistency. They are not soluble in water or sparingly soluble in water, but they are soluble in organic solvents like ether, alcohol and benzene. When you talk about the types of lipids or fats, they may be of three types: simple lipids, compound lipids and derived lipids. In this chapter, we are basically dealing with simple lipids. Mainly living organisms have simple lipids in them. They are esters of fatty acid and an alcohol. Hope you remember ester formation you learned last year in class 10 carbon and its compound lesson. There ethyl alcohol and ethanoic acid you combine to derive the corresponding ester. So here also fatty acid, so it is an organic acid, it esterifies with an alcohol. So natural fats and waxes are examples of this esterified fat. So usually in our body, these fats are chemically fatty acid. The acid involved here is, there we learned the example of ethanoic acid and ethanol. Here the fat is fatty acid and the alcohol is glycerol. In order to understand the structure of lipid or fat, we should know the component structures like fatty acid and the alcohol group. So here fatty acid is having a structure called a RCOOH group. You know, COOH is a functional group which represents acid, right? COOH group. To that one R group is here. This COOH group is called the head of this, and this makes it hydrophilic. Philic means what? Loving, water loving end. But this R group is having CH3 group, that is this CH3 is called what? Methyl group, or it can have C2H5 group. What is that? Ethyl group. To that, CH2 several times, n times CH2 will be there. For example, CH3, CH2, n times COOH. So this is the nature of one acid, fatty acid. So this group is the head part, which is the carboxylic group, but this part keeps varying. This is the R group. This part is a long hydrocarbon chain which makes it hydrophobic, means water hating. So examples, if you see, we have an acid called a palmitic acid which has CH3, CH2, 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 like this. How many times you have to write? 14 times, then COOH will come. So we can change this as 14. So total how many carbon atoms are there? CH2 14 are there. So 14 plus this carbon 15, carboxylic carbon 1. So total 16 carbon atoms are there in palmitic acid. You have to remember this. Palmitic acid has 16 carbon atoms. One more example you have to study, that is arachidonic acid. Arachidonic acid has total 20 carbon atoms including the carboxylic carbon. So usually this number of carbon atoms can vary from 1 to 19 except this COOH in the R region. Now you learned about saturated and unsaturated hydrocarbons in your previous classes. So you know that if it is saturated, there is only single bond existing because enough number of hydrogen are there. So single bond exists between this CH2. So it is called a saturated. For example, palmitic acid is a saturated acid, or a butyric acid are saturated. But there can be double bond also in between. Then we call it as unsaturated fatty acid. And based on the melting point, we can classify them into fats and oils. Usually if they are solid at room temperature, they are called a fat. Example, you take ghee or butter or coconut oil. So if you keep it outside during winter season, they usually tend to be solid in nature. Whereas the sunflower oil, gingerly oil, corn oil, olive oil, all these oils, even if you keep them inside the refrigerator, they don't solidify. They are unsaturated fatty acids. They are called oils, whereas saturated, which can solidify, are called fats. So we saw the acid part. Now which is the alcohol found in lipids? It is glycerol. So let's see the glycerol. Glycerol is a trihydroxy propane. So you know propane, last year you learned how many carbon atoms are there? 3, right? Prop means 3. CH3, CH2, CH3. But in this each one, if I am giving a hydroxy group, OH group, then it becomes alcohol. You know that OH is the functional group for alcohol. So if it is CH3, CH2, CH3. Here one hydrogen I am removing and adding one hydroxyl group. Then here also I am substituting with a one OH. Here also one OH. Now we can call it as tri means what? 3-hydroxy propane. Okay. So this is nothing but the glycerol. So we learned the acid part, we learned the alcohol part. But now we know that in order to get the simple lipids or fats, they have to esterify. Esterification reaction is removal of water, the acid and alcohol combines. So in this case, we will see how it is combining. This is the carboxyl group RCOOH. We know that R is the hydrocarbon group, this is the head part. So from here, one hydrogen and from here one OH will combine. So as a result, what is going out? A water molecule will go out. So the same way, so this is called a monoglyceride. That means when one fatty acid is esterified with the glycerol, that is called a monoglyceride. Now I will write one more. Now here one more molecule is going. Then I can rename it as diglyceride. So if I add one more, now you will tell me what is it? Exactly, triglyceride. So triglycerides means 3 fatty acids esterified with a glycerol. Esterification reaction means what? Removal of one water molecule. So what is the structure of a triglyceride? We can write CH2O, C, double bond O, R, because water molecule has been released. Now next CH2O, C, double bond O, R. CH2O, double bond O, R. Okay, so this is the structure of a triglyceride. So now these kinds of molecules where only fatty acid and glycerol are there, they are called simple lipids. Sometimes there can be other compounds to it like sulfur, phosphorus, nitrogen, then we call it as compound lipids. In compound lipids, example is a phospholipid. Phospholipid, from the name you can understand what is it. Phosphoric acid group or a phosphate group is attached. Here that phosphate group is phosphoric acid, then it is making our cell membrane. Because in cell chapter you are learning about the cell membrane structure, Singer and Nicholson model. Lipid bilayer is there. There we have phospholipids. Phospholipids have phosphoric acid attached to it, and the main one forming our membrane is a lecithin. You have to remember this name, very important, lecithin. Okay, now another one, maybe carbohydrate is attached, then we call it as glycolipid. Now what are derived lipids? If the simple lipids and compound lipids they combine, then it is called derived lipids. These derived lipids examples are steroids, cholesterol, carotenoids, etc. Now coming to some uses of lipids, just to know, lipids are mainly the storage products in plants and animals. Fat is stored in large amounts in hibernating animals as a source of energy during hibernation. And lipids can dissolve the vitamins like A, D, E, K, which are water insoluble vitamins. And waxes on the surface of animals can act as waterproofing agent. The same way plants also have cuticle or waxy coating over the leaf which prevents transpiration. Phospholipids, glycolipids and sterols are helpful in forming the cell membrane of the organism. And cholesterol is a raw material for our sex hormones and adrenocorticotrophic hormones. And in animals there is a fat layer under the skin which protects them from harsh weather. Now we are moving on to another biomicromolecule, that is nitrogen base. So where do you find nitrogen base? We know that our genetic material is DNA, or some viruses have RNA. So this DNA and RNA are called nucleic acids. Nucleic acids are biomacromolecules. They are made up of many nucleotides. Each nucleotide has got three components: phosphorus, sugar and a nitrogen base. Of this, these three together called nucleotide. If you remove phosphorus, sugar and nitrogen base together called a nucleoside. We are going to learn about the nitrogen bases present in this nucleotides. So nitrogen bases are classified into two categories: purines and pyrimidines. Whether it is DNA or RNA, four nitrogen bases are found. So the purines are basically double ring structures. They have two types, adenine and guanine. Whereas pyrimidines are also two types, actually three types: cytosine, thymine and uracil. If it is DNA, thymine is present. If it is RNA, uracil is present. So in DNA we have adenine, guanine, cytosine and thymine, whereas RNA has adenine, guanine, cytosine and uracil. Thymine is absent there. Now when we add this nitrogen base, and these pyrimidines are single ring structures, always a purine base pairs with one pyrimidine. Now if you add one sugar also to this nitrogen base, it becomes nucleoside. Sugar can be deoxyribose sugar, which is present in DNA, or ribose sugar, which is present in RNA. Both are pentose sugar, means 5-carbon sugar. So if adenine is combined with a sugar, we call it as adenosine. Guanine is combined, we call it as guanosine. Cytosine plus sugar we get cytidine. Thymine plus sugar is thymidine. But these are ribose sugar. If it is deoxyribose sugar in DNA, we rename it as deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine. Or if it is RNA, uridine. There is no need of telling deoxy because it is not in DNA, it is only in RNA. Now to that one phosphorus is attached, we get nucleotide. So let's name the nucleotide. Phosphorus means it's phosphoric acid, so it becomes acid. Adenyllic acid, guanylic acid, cytidyllic acid, thymidyllic acid or uridylic acid. So this is how the nucleic acids are formed. So many of these nucleotides are combined to form DNA or RNA. Hope you understood all the topics well. If so, please like, share and subscribe to my channel Biology My Passion. Thank you for watching.